My name is Anna Thelen. I am 23. I'm working as a chemistry technician. I work with medicine, mostly pills, capsules, tablets. I break them down into a liquid form and mix them with different chemicals, and we test them. I really like it. It's really interesting to me because I can do things with my hands and do experiments and see things like colors change and different reactions.
We are a quality assurance lab. We are making sure that what it says on the bottle, says on the labels, is what's actually in it.
We have a pretty large microbiology lab that tests for safety of different food products and the quantity and presence of active ingredients in dietary supplements.
I went to Rochester Institute of Technology. I graduated with a Lab Science and Technology Associate's degree. I can work with biomedical, environmental science, biology, nursing, anything like that.
When I found out that Anna was deaf, at first I was like, I don't know how well she'll be able to understand us with all this scientific lingo.
My major concern was that people won't want to interact with me because they don't know sign language or they don't know how to communicate with a deaf person. So I have to approach them and show them what to do.
Teach us so that we can sign from across. Having that extra focus on effective communication, you start to realize that you really do need to rely on each other to be able to get everything accomplished that needs to be done in a day. You've already checked that your standard amounts were updated properly? Anna's a hard worker. She'll do anything that I ask of her. She's reliable. She's a team player. I don't think we could do what we do every day without having a good team environment.
You know, you're over there and I'm here and I'm like... It's nice to be able to speak in her language.
She shows us a few signs here and there, and we try to pick up on as much as we can. So we've been slowly learning.
When I came in here first, I was a little doubtful about how it would work out, and maybe not sure about how hearing people would look at me as being a deaf worker. But after a little while, they got used to the idea of having a deaf person be here.
As scientists, our first job is to learn and to understand things. And someone who is deaf is just another thing to figure out. So me and Brittany jumped on the chance to be able to learn sign language because it's fun.
The opening and closing injections, are they good? Okay, good.
I like employers to keep an open mind when working with deaf people and be willing to learn new things, new ways to communicate, such as learning a little bit of sign language themselves or learning how to gesture.
Lab sciences are a field that need more diversity, and you need your mind first, not your ears.
I really do like this job, and I like working here. I can imagine myself here for 5-10 years, and I could see myself becoming a supervisor with the department.
Okay, so good morning, everyone. You're awfully quiet this morning, okay? And look, we've made it. It's Wednesday morning. We've made it through the majority of the conference. A couple of things. Hopefully you've met new people. Everybody made at least one or more new contacts? Yes? Come on, a little noise, Okay, good. All right, so one piece, just kind of a general housekeeping, is we've got the last showcase following this presentation this morning. So I know some people have to leave. They've got a flight that they can't get out of. But if you can, please attend the showcase. Think about the people, if you've already showcased, think about the people that came by your booth and talked with you. So please do the people that are in this last showcase the courtesy, and especially our students, the courtesy of staying for the showcase if there's any way that you can do that. So it's my pleasure, first of all, when Dave got up and he said, hey, I get to introduce Celeste's boss, remember? Okay, so now I get to introduce Dave's boss. So my first chore here this morning is to introduce to you Dr. Evan Height, who will be getting up in a minute to give some greetings from the National Science Foundation. He is the division director, just recently made permanent, of the Division of Research and Learning in Formal and Informal Settings. We really love acronyms, right? It's DRL. So he started as a program director in DRL and came to the National Science Foundation from one of the newest UC campuses, the University of California in Merced. He was one of the founding faculty at the School of Social Sciences, Humanities, and Arts there. His history is, as far as his research area of interest, he's a psychologist and cognitive scientist. So he's got a really kind of unique perspective, at least from my standpoint, and probably a lot of us thinking about real disciplinary matters. Evan is one of those people who is thinking about things like reasoning, learning, and memory. So I will invite Evan to come up and give you greetings from the National Science Foundation.
Okay, good morning, everybody. Thank you, Celeste, and thank you also to Dave Campbell. It's so great to work with both of you, and you've made so many great accomplishments working with the ATE program. I also want to thank everyone here for just what you've contributed as being ATE investigators. I could tell you that at NSF, ATE is a program we really treasure. We really celebrate this program. I would like to think that all of our research and education programs at NSF contribute somehow directly or indirectly to the STEM workforce, but there's a nice, clear, and transparent relation between the work that you're doing and building the nation's STEM workforce in a particularly important sector of the workforce. And we really value that. Personally, I'm really impressed by the partnerships with industry that you've built up over the years. It's just really nice. It's something we are quite proud of, your work at NSF. I'll just mention, since I come from DRL, and that there won't be a test, but that's a Division of Research on Learning, that we have other programs that we also work on. So ATE is something we work on with the Division of Undergraduate Education, but I'll just mention a few other programs. We have a program called iTest, which is Innovative Technological Experiences for Students and Teachers. This, in some ways, is a counterpart to ATE. It's a younger age group. They're not necessarily getting directly prepared for their workforce, but we're trying to build up STEM interest in all sorts of STEM fields. We also have a K-12 program, the signature K-12 program at NSF is called DRK-12, and we have an informal program for all sorts of informal STEM learning settings called AZEL. And I'm mentioning these just because you are successful investigators for ATE, and I encourage you to look at other programs at NSF, including iTest, DRK-12, and AZEL, because we would love to have more great proposals from successful investigators like yourselves. I personally would be interested in seeing more partnerships in industry in these programs. The iTest program, right now it has about 40% partnerships with business, but just in general, it's something we do like to see at NSF. And if you'd like to learn more about iTest or AZEL or DRK-12, please feel free to come find me or Dave Campbell after the meeting, and everyone knows Dave. And we'd be happy to talk to you more. And thank you for having me here today. It's very nice to be back.
Okay, so thank you very much, Evan, for that welcome from NSF. And it is, actually, most of you know about the informal science program. Anybody who has children knows about it, right? Anybody ever seen the Magic School Bus or Bill Nye the Science Guy? And you get to the end of some of those shows and it says, there's the NSF logo and it says funded in part by the National Science Foundation. So most of you know about some of those informal science programs, but there's also citizen science, which is very, very interesting, where the general public is actually gathering data that's used and accumulated around a specific topic area. So definitely find Evan or find Dave. I know a little bit, not too much about some of the programs, but I can always direct you to another program officer. So it's now my privilege to introduce our speaker for this morning, Dr. Jen Kim Montclair. And she's going to be talking about STEM pathways towards research and innovation and lessons learned. And there's a lengthy bio in the book, okay, but I'm going to just give you a couple of highlights. One thing that particularly interested me, being a sort of virologist, cell biologist, molecular biologist type of person, was that her research area is looking at engineering proteins to mimic nature and then sometimes develop a synthetic protein that works better than what you find in nature, which is pretty interesting. So there's a focus there on biosynthetic machinery, which crosses not only biology, but I'm hoping all the people that are interested in engineering might think about this as a particularly interesting area as well. If you look at the extended bio, you're going to see that she has numerous awards. She is an associate professor in the Department of Chemical and Biomolecular Engineering at New York University, and there are several other appointments there as well, but I want to give her time to do the presentation. So Dr. Montclair, I'll turn the stage over to you.
Hello. Thank you, Celeste, for that nice introduction, and thank you, Ellen, and all the organizers for inviting me to give a talk here. Before I begin, I'd like everyone to stand up and stretch, because whenever I'm sitting at a conference, I usually start slumping in my seat, and then I get tired, and then I forget, then I'm in a conference, and then I might doze off. So if you stand up and stretch and then kind of get your adrenaline going, hopefully you'll be able to manage to stay up for the rest of my talk. Okay, so now you can sit down. I'm actually new to this conference, and I'm excited to be here because I have an interesting pathway, and I'd like to share that story with you. And so I'll begin. Okay, so today I'll be talking about, oh my gosh, I have two screens, sorry. Today, I'll look right here. Today I'll be talking about several things. I'm gonna talk to you about my own experience or my journey from K to 20 and the mentors that have helped me through the way. I'll also then talk about the actual research that we do in my lab. We do biomaterials research, and specifically I'll look at the biotechnology and medical applications of that research. There's other applications and other types of research that we do, but I thought for this conference it'd be better to focus on a couple of things. Then I'm going to be talking about my passion in engaging the K-12 STEM population outreach and technology that we've developed to help that. And then I'll talk about entrepreneurship, increasing women and diversity, and then my lessons learned for those of you who need some lessons at the end of the meeting, and you can take them with a grain of salt as you proceed on with your day. So let me tell you about my background. So I didn't start here in the United States. Actually, my parents started in Korea, and then they emigrated to the United States. And then I was born here in the Bronx in New York. And my family was not just my parents, it was actually my grandparents and my parents. So both my parents, my mom and dad, they work full-time jobs, and so I was raised by my grandmother, my brother and I, so my brother is over there as well. And these were important people, at least my grandparents were important people, at least in my life, and helped me, actually was one of my first mentors in my experience as a scientist. So I was a little kid, I was short, and because of my height, I tend to look down on the floor because there are interesting things around me by the floor. And I was really fascinated by ants and bugs when I was a little kid. And so I would watch them, I would follow them around, see these little ant holes, and then stomp on them occasionally, maybe dissect them. But what I recognized as a kid, or as I look back, is that nature is full of curiosities that I could study and explore. And I really, really loved just spending time outside and trying to find and capture bugs and do all sorts of fun stuff with them. So these were my friends, and I started to build a network starting with my little ant friends. However, what was really important was as I was going through school, especially in kindergarten to 12, were my teachers. My teachers really, really guided me into the sciences because I seemed to like it, and they continue to support that. And in particular, I'd like to focus on my high school teacher, Mr. Skolar, shown here. He was the one that started my spark and interest in chemistry. So he would have these intricate chemistry experiments in high school where literally we would blow things up and just make all sorts of crazy concoctions related to chemistry. One experiment in particular that really convinced me that I should study chemistry for the rest of my life was he took, and it was a fun experiment. He took a funnel, piped it into a gas line, into a bubble mixture, blew a bubble, the bubble floated up in the air, had a candle, and then lit the bubble, right, popped the bubble, and it lit on fire. And we had this huge, like on the ceiling, fire. Of course, of course, what happened was the sprinkler system went on. We had to evacuate our chemistry classroom, our lab, but I thought that was the most fabulous thing in science ever. And I was completely hooked with science from then on and thought chemistry was the way to go. And so he was the one who really sparked me on to this, to chemistry in particular. And then what happened was I went to Fordham as an undergrad. Again, I'm from the Bronx, that's where I went. I had mentors, Jim Chacho, Saha, and Bob Beer. These were my mentors. I did research in their laboratories. I also started to become interested in biology, so then I decided I'd go to my neighboring school. This time it was Albert Einstein College of Medicine. I worked in the genetics department under Raju Kuchalapati, and so I had this love of chemistry and biology. And so after getting my undergraduate bachelor's degree in chemistry, what happened was I wanted to marry both my love of chemistry and biology, and so I went to Yale and got my PhD under the first female chemistry professor at Yale that was tenured, Alana Shepherd. And she was actually my first role model as a female. All throughout my college years, I had mostly male professors that were great, and I'm so grateful for them. But I was also grateful to see someone who was like me, who was a female, doing research in a laboratory. And so I did some studies on generating transcription factors, engineering proteins, and I had a wonderful time there. I learned from her that hard work is hard work. You have to actually put in the work. And we produced a number of papers from that research. I subsequently then went to Caltech, and I worked as a postdoctoral fellow in the laboratory of David Terrell, shown here. He was interesting in the sense that I was able to take my knowledge of proteins and then engineer them using evolutionary approaches to make them more stable and more effective. And what I learned from him was that I don't need to sit in my own discipline, that I can actually go and interact with other laboratories and other professors and try to develop new ideas. And so collaboration became an important point in my life when I went to work with Dave. And so where did my education take me? I started at Fordham, went to Albert Einstein, which was just, not too far away. Then went a little away from New York to Connecticut, to Yale. Then went all the way across, actually drove across from Connecticut all the way to California, and then came back to where I'm at as a professor at NYU Tandon School of Engineering. So these were my multidisciplinary networks, and I had, as I started my career at NYU, I continued to grow a network of colleagues in various disciplines, in medicine, in electrical engineering, in mechanical engineering, physics, dentistry, computational biology, because I really think that in order to achieve new and interesting science, you have to be willing to interact in a multidisciplinary group, and it's actually, you learn a lot more, because as an educator, you're a lifelong learner, and it's always wonderful to learn from other disciplines. So, right, so this brings me back to the little ants. So that was my inspiration when I was a little short kid, still short, but not so little. And recognizing that nature is full of curiosities that I can study and explore. And using that, I became interested in nature, like through the billions of years of evolution. Nature has all of these examples, like these ants. If you look at leaves can harness energy from light. If you look at this bacteria shown here, it's a magnetotactic bacteria. This bacteria can crystallize iron oxide nanoparticles that allows them to sense the electromagnetic waves and help it swim. And then if you look just down at your hands are an awesome source of materials, because when you cut your hand, it's able to self-repair. So all of these examples in nature have principal components, and in my view, because I'm a bit biased because I like proteins, the building blocks that all of these examples are based on proteins. And these complex functions that these organisms can achieve are achieved through these proteins, which are shown here. So my lab is interested in engineering artificial proteins with emergent properties. So if we take a protein is comprised of 22 amino acid building blocks, and there's a structure shown here. And it has defined secondary structure like alpha helices and beta sheets that allow these proteins to function the way they do. And so what we do in our lab is we take that information from nature, we take our molecular scissors, and we cut these proteins up. And we isolate elements of these proteins that we think would be beneficial for our purposes. And then we piece them or tape them in ways that nature hasn't thought of, in the hopes that we can make new types of proteins, artificial proteins, that can have new types of function. And on top of that, what we're able to do is we're able to integrate non-natural amino acids. These are building blocks that nature has never, ever thought of, like fluorinated amino acids, like photocross-linkable amino acids. And the whole idea is to integrate that into our artificial proteins, make these constructs, and then hopefully make them useful for a particular application. And so that's what we set out to do in my lab. And so the way we make our artificial proteins is we do program our bacteria. There are our living factories for making our proteins. And so we figure out what gene we want to integrate, and we take that gene, integrate that into a plasmid, and then use our bacteria to produce the protein that we want. This is often called synthetic biology, and then generate these new proteins that hopefully nature hasn't necessarily seen, but can be integrated and used with nature so that we can help control some processes that might be bad in medicine. So why are we interested in making new types of materials using proteins? So if we look at the materials that are used in, for example, medicine or biotechnology, much of them are synthetic. for example, there's poly-L-glutamic acid, or PLGA, which is a polymer. PLGA is a polymer. And the synthetic polymer is also PMMA, or polymethyl methacrylate. It is polydispersed. If you look at the mass spec, you see this wide dispersity. PLGA, which is L-glutamic acid, we can genetically engineer that construct, and we can genetically engineer that into defined discrete molecular masses so that we get monodispersed constructs. Moreover, because we have secondary structures, access to alpha helices, beta sheets, we can then piece them together and make new types of materials as shown here below. So the question that my lab asked is, can we construct intelligent materials? So many of you have your cell phones, your iPads, or your laptops. These are machines that can sense you and that can respond to you. And so using that principle of sensing and responding, can we do that, or do that on the nanoscale, on a scale where we can engineer proteins that can then be used to help the human condition in terms of disorders, et cetera? So that's the question we set out. And in order to do that, we made a couple of biomaterials, and I'm just highlighting a couple of them here. And in particular, they combat 3 different disorders. One I'll be talking about is breast cancer. Another is osteoarthritis. And then the final one is on diabetic wound healing. And we're all doing this through nanomaterials that we've constructed on the protein level that nature has never devised before. And these are nanoparticles shown below in that arrow, or nanofibers, as you can see in the middle picture. So let me talk to you about metastatic breast cancer. So there are 40,000 deaths annually in the US due to metastatic breast cancer, or MBC, with an average survival rate of 18 to 24 months. And there is a chemotherapeutic that is used currently, and it's called doxorubicin. It can reduce cancer, but it is toxic. It causes severe vomiting, hair loss, alopecia, among other nasty side effects. And the mortality rate for breast cancers, at least metastatic ones, are 99%. So the chemotherapy shown here is a molecular, if you're a chemist, right, structure of doxorubicin shown here. It's very good at killing cancer, but they have the following challenges. One, it's insoluble, right? If you look at the structure, it's very hydrophobic. They can break down readily due to instability. 2, they're non-specific killing, right? And because they're non-specific, they can kill healthy cells. And 3, they're rapidly cleared from your body. So then the therapeutic effect is limited. So some of the solutions as engineers, as we think about this, is to encapsulate this. So if we encapsulate the small molecule, you can avoid solubility issues and the breakdown. You can then, if you develop something to encapsulate them, you can then add active targeting by actually heat as a mechanism, or adding specific tags. And then if you make them on the nanoscale, if they're nanometer size, you can exploit this phenomenon called enhanced permeation retention, which happens in tumor leaky vasculature, so that in tumor leaky vasculature, you have more resident time of the material in the tumor, so that hopefully it doesn't get rapidly cleared. So what do we do, and how do we make our new materials? So we use a couple of components in making our materials. Here are the building blocks. So you see above is a tag that can template gold nanoparticles. We have an elastin domain. This is found in tissue. It allows your tissue to be elastic, and it has conformation that allows it to self-assemble and coacervate. The C domain, this is a domain, a coiled coil domain that self-assembles into a pentamer. And what's nice about this C domain is that it has a hydrophobic pore that can bind small molecule. So hopefully that can bind our drug. And then you can add cancer peptide targeting domains, and then F is actually fluorine. So remember I had said that we can incorporate unnatural amino acids that nature has never decided or ever thought of. And so fluorinated amino acids are things that nature never had thought of. And so we started off making these materials. We took two components in the first case, where we took the elastin domain and that coiled coil domain, fused them together. They assembled into these nanoparticles, and these nanoparticles can be loaded up with small molecule to help with delivering the cancer therapeutic. What we did subsequently is we took gold binding or gold templating tags and templated gold nanoparticles, which then enhanced the binding of the small molecules, in this case, the cancer, anti-doxorubicin, and curcumin is another one. And then these were incubated with breast cancer cells and shown that it can deliver the payload to the breast cancer cell. And then more recently, what we've done is we've integrated fluorinated amino acids in a similar amino acid consistent with leucine. So in place of leucine, what we did was we incorporated what is called trifluoroleucine, which is fluorinated version. And so because it's fluorinated, we can actually image it, but we can also enhance the binding to small molecule, target it to cancer cells. So we can encapsulate the drug, target to cancer cells, and these are nanometer-sized particles. And so if we look at the fluorinated case, we see that we can generate these micelle-like structures that are in the nanometer size. These can have these fluorines, and then using fMRI, we can now image these nanoparticles. So now that we can increase or integrate the drug into these nanoparticles, we can now image them non-invasively so that you can monitor the efficacy of the therapy non-invasively over time, and you can eliminate invasive biopsies. And so that's what we've been doing in our lab. The second story has to do with osteoarthritis. If you look at a healthy joint and the osteoarthritic joint, you can see damage occurring where your bone is starting to show through, the cartilage is degrading, and it causes many, many Americans a lot of pain. Here it's a common chronic condition of joints affecting 27 million Americans. It can affect joints, but it occurs mostly in the knees, hips, lower back, small joints, and fingers. And so this is something that we're trying to treat. My colleague at the medical school has identified a drug. It's called BMS 493, very interesting name. And it's a hydrophobic small molecule that's actually very light sensitive, so it degrades. So while this is proven to be effective in treating osteoarthritis, it degrades, so you have to continuously inject this in order to have its therapeutic effect. So the challenges is that it readily breaks down due to instability, and it must be injected multiple times. Our solution is that we can encapsulate it to avoid that breakdown and hopefully slow in its release so that you don't have to continuously inject in the joint. So what do we do in this case? In this case, we've developed a nanofiber. This time, we're only looking at that coiled coil domain. These coiled coil domains, remember, forms a pentamer, and it can bind small molecule. But what we found was that it can also self-assemble into nanoparticles. When we add the BMS 493, it actually forms these fibers that are stable, that actually slowly release the BMS 493 so that we can just use a single injection and use sustained release to treat osteoarthritis. So that's another example of drug delivery. And then finally, the final example has to do with diabetic wound healing. And so diabetic wound healing, it affects 29.1 million people, and 15% of these patients suffer from chronic ulcers. And chronic ulcers are these non-healing wounds. So these are a challenge to cover with soft tissue, and they represent one of the leading causes of lower extremity amputations. So this is something that we're challenge as engineers to develop a solution. And so in this case, what we're looking to is treat this with siRNA delivery. So my collaborator at the medical school has developed or has identified a gene, KEAP1. KEAP1 has been known to, when you silence that gene, to accelerate wound healing in diabetic patients. And so what we wanted to do was we wanted to develop something that could actually deliver this siRNA to the site of the wound. And so what we did was we looked at our C domain, right, that coiled-coil domain, and that within itself, while it cannot bind siRNA because it's negatively charged, we can actually re-engineer this system so that we can have its surface supercharged to bind to the siRNA. And so what we did was we made it supercharged. That's why it's going from red to blue. That supercharging allows us to have it bind to the siRNA. And then once we form that, what we call proteoplex particle, so it forms this particle, we then add lipids to generate what is called a lipoproteoplex, and an image is shown there. It's still in the nanometer range. And that is what we're using to deliver genes to wounds. And so shown here is a mouse model of a wound where it's untreated diabetic wound on a mice. And then when we add our lipoproteoplex, this is called DNC, CSP, with the siRNA for the KEAP1 gene, we're able to reduce that time of wound closure, accelerating it. And so this is another thing that we're working on in terms of materials for medicine. And so the question we asked in my lab was, can we construct intelligent materials? And the answer is yes, right? So we're able to do that. And we're continuing to do that in the lab on other disorders and many other things. So that's a little bit of the research that we do. And so then I wanted to reflect, well, why I love what I do. I love what I do not only because we can do cool science and engineering feats, but I love what I do because I'm interacting with the next generation scientists and engineers. These are the students that have been doing the work. I did zero of the work. I mean, like all the experiments were done by my students, and shown here are some of them. And what I find really awesome is that I'm continuously learning from them. Many of the ideas that came up in the series of experiments that I've shown you have come up from these students, these young, brilliant scientists and engineers. And what I really like is trying to understand from them what they want to do in their lives, what their pathways will take. And that is multitudinous. So I have students who are gone to industry, gone to government, they're gone into consulting, academia, some are entrepreneurs, and that's really amazing to me. And I want to continue to do that. So I had also told you that another passion of mine is K-12. And when I showed you my valuable list of mentors and supporters through my career, really the most important one that got me into this chemistry pathway was my high school teacher. And so K-12, I believe, is a crucial time. And so for me, it was actually these hands-on experiments that totally blew my mind away, got us into a little bit of trouble, but still was really fun, that really compelled me to go into this field. And so I have, since 2008, been working on outreach programs, just developing it on my own. And so we started this outreach program called the Chem Biotechnology Lab. This is a program in collaboration in Brooklyn with the Urban Assembly Institute for Science and Mathematics for Young Women. And so here's a subpopulation of the group that we had taught several years ago. We continuously teach different groups. And so the idea I had come up with was, well, many of these students don't see themselves in farther down in education. And so maybe if they get connected to undergraduates, they'll be more willing to go into college. And maybe they'll see me as a professor and they'll be more inspired to pursue higher roads of education. And so what we developed was a mentorship program where I have 2 undergraduate students who we work with a teacher in a classroom and develop modules that are either hands-on or virtual so that students can learn chemistry, for example. And so as part of that, we did a number of different types of learning modules. One we developed was an app called Lewis Dots, and it helps you form molecules. And we use that to help the students learn about chemistry and make learning a bit fun and engaging. That got a lot of attention. We published a number of different papers on that. And we were highlighted in a New York technology meetup where we showed our app to everybody. And then all of this got my student, my graduate student, Carlo Uvienko, very interested in developing a company based on this. So we were encouraged to apply to a program sponsored by the NSF called the I-Corps program. It's a startup boot camp using Lean Launchpad. And Carlo, he was my PhD student in biomedical engineering at the time. He was the entrepreneurial lead. He took the reins and he said, I wanted to do this. And so I said, okay, how can I facilitate you and get this done? And so we found this program. We had to enlist an entrepreneurial mentor. In this case, we got Andrew Cohen, who is the CEO of Brainscape. And then we applied to the NSF I-Corps program, and we started our journey. We did 108 customer interviews. We pivoted from that Lewis Dot apps to a more larger platform called Lab Lessons, which helps teachers develop pre-labs to help students prepare for the labs. And then we started a company called In-School Apps. So the lab lessons is a pre-lab. It's essentially pre-labs to prepare students for labs. Our pilot has been with, again, we're located in Brooklyn Technical High School, chemistry class and teachers. And so we've been working with them and developing this pre-lab out with a component where it helps grade for the teachers. And so we're hoping to, again, inspire the next generation to love STEM. So that brings us to another component, which is entrepreneurship, at least in my path towards that. So as my students and I were doing all of this stuff with startups and entrepreneurship, I became interested in looking at our school and looking at the landscape of our school and seeing what our school provides in terms of innovation, invention, and entrepreneurship. And so we found that there's a lot of things going on in the school. I developed a meeting with several faculty within the engineering school, and we found that we had incubators that were located within the institution. There were various competitions that were undergoing, focusing on hackathons, focusing on capstone competitions, et cetera. And then there's also clubs, student clubs that existed. So we had various student clubs, all focused on entrepreneurship. And finally, there was a newly developed greenhouse where students would talk to one another and develop ideas, and it's student-led and student-run. And so all of these components that were going on at our school, at the engineering school, led us to believe that this is something that we should, as faculty, be trying to actually shepherd. And so we realized there was a lack in curriculum that's focused on innovation and entrepreneurship. Most of the curriculum in engineering schools focus on engineering classes and very limited time for extracurricular activities. And so what we decided to do as a faculty of that group was start to focus on programming or curricular development that would integrate some of this innovation and entrepreneurship into the classrooms. And so that's where the green circle, that's where we came in. So we then form this Convergence of Innovation and Entrepreneurship Institute. Our mission is to increase diversity in STEM entrepreneurship and provide guidance as well as resources to STEM innovators to start up. We developed several things. So we started in about 2014. One of the first things that we did was we developed a capstone competition. Our initial capstone competition started in 2016. We were applying for funding. We applied for a VentureWell grant, a pre-capstone course, and that got funded in 2017. And then we got funded for the National Science Foundation I-Course Sites program to train teams of students on entrepreneurship just in 2017, and had our first cohort this summer. We also, as part of that, as diversity and inclusion is a mission, we developed an e-inclusion network through our CIE, where we have various students and entrepreneurs who are women or underrepresented, building a network for them, so for support. So we started our first NSF I-Course Sites program at NYU. Again, our focus was on increasing diversity. And so we had a hypothesis, and this was actually written in our grant. Our hypothesis was that STEM entrepreneurship that is geared towards social goals will be more appealing to all students, and women in particular. And so we developed this course, this NYU I-Course Summer. It's a 4-week program that uses that lean launchpad methodology to show the teams how to develop STEM ideas into valuable commercial products. And the benefits were that they would be provided funding up to $3,350 to produce prototypes for successful participants. They would get training in lean launchpad methodology. They would be partnered with technical and business mentors, and then they would do their own validation of customers and hopefully identify a minimum viable product. So what we did when we set out our program, set out doing our program prior to the actual summer program, we actually set out a survey for the students. And what we were trying to identify were what are the factors that impact diversity in STEM entrepreneurship. And so what we were interested in doing as part of this survey was not only to help market our ideas, right, to say, hey, we're having this NSF I-Core program, but also understand diversity more generally. We got 221 responses, 127 finished of those 221, so 57%. NYU's a huge school with various different separate school siloed. So we're part of the engineering school, but there's arts and sciences, there's the medical school, the dental school. And so we had 7 schools of NYU being represented, but 92% were from our school, the Tandon School of Engineering. My guess is I had sent out the email, so they know who I am at the engineering school, but everywhere else it wasn't known. They're mostly STEM major, 96%, and then mostly male, 58%, and undergraduates. So it was actually a survey for not only undergraduates, for graduate students and postdocs. So 61% were undergraduates. So what we did was we surveyed them, asked them a bunch of questions, and here are some points that we identified. Why do people become entrepreneurs was one of the questions. So women, most of the women highlighted that they make a positive impact, and you can work by your own rules. The men, at least in the survey, was focused on making money, and you can work on by your own rules. Some of the quotes, this is one quote, students are teeming with great ideas, this is from a student, and just need some mentoring and a little push in the right direction. These students are the future, and what they create today can impact the world they will live in tomorrow. These are beautiful quotes. So our hypothesis was somewhat affirmed, right? STEM entrepreneurship that is geared towards social goals will be more appealing to students, in particular women. And so with that, we heavily recruited. We did lots of info sessions, panels with focusing on women and diversity. And then we had our first class. Our first cohort this summer was 58% women and underrepresented minority team leaders. So that was a really happy, at least outcome for us. And we'd like to continue learning and continue focusing on that. And here's one of our team. It was like an all-women team, Lost Bites team. And so that's a little bit of the entrepreneurship component. And then finally, I have lessons. So you have these lessons, take them with a grain of salt, but these are the lessons after going through all of this and sharing you my life story. So, important lesson number 1, challenges from naysayers. All right, so you'll have people who will constantly tell you one of two things. You can do it, that's an awesome idea, or you can't do it. That's a terrible idea. So my solution to that is to have selective hearing and just put your nose to the ground and work hard. So here's your selective hearing aid. Tune down those naysayers, focus on the positive, and focus on the support that will get you through the day. And then consult your mentors and colleagues and supporters, and put your nose to that ground and work hard. And so that's lesson number 1. Lesson number 2. As I've shown you in my story, I had a lot of help to get to where I'm here, at least to this point. So I sought mentors, and I think mentors were the people that allowed me to get to where I am. So you want to surround yourself with people who you would like to learn from, who you think are doing the smartest things and the coolest things in the world. And you want them to lift you up and help you. And so seek your mentors. I had all of these people that were behind me, that made me continue on my path. Without these people, I don't think I would be standing here and speaking to you at a conference like this. Important lesson number 3, seek help. If there is something you do not know how to solve, find people to help you solve that and collaborate. I have found that one type of discipline cannot solve the complicated problems of drug delivery and medicine. I need help, and I'm happy to say I need help. I go out to my colleagues and ask them, what do you think? And they provide input, and we collaborate. And the only way we can effectively make advances in science and engineering is to collaborate and do multidisciplinary work. And that's my perspective on it. And diversity, in particular, diversity in perspective always helps to identify new solutions. And I've seen that case by case by case in all the different problems that I had to solve. Important lesson number 4, fear of failure. So yes, there is a word as failure, but I don't actually view it as failure. I view it as a continuum of learning. So what people normally view as a failure is what I view as a step towards understanding. And it's a piece of information that I've learned. And so do not think of it as a dichotomy that this is a failure and this is a success. Rather, if you think of it as a continuum and you're learning as you're going, you're actually gaining knowledge, and that's a positive thing. Important lesson number 5, give back. Pay it forward and help others, right? So someone helped pull you up. Now it's your turn to help pull other people up. Inspire that next generation and make that world a better place. So these are the people that I work with. These are the people that I have to pull up, and so are these. And so hopefully we can make the world a better place from doing so. And then that's it. So all I have to do is acknowledge there's a bunch of people that I put their names on the slides in terms of research. These are all the students and collaborators. I have to thank all of our funding, the National Science Foundation, Department of Defense, Army, Department of Energy, NIH, Dreyfus Foundation, Teagle Foundation. And if you want to follow us, you can follow us on Twitter at Montclair Labs or at JK Montclair. And then that's it, and I thank you for your attention. Oh, right. I'm open for questions.
Okay, and I'd like to just say one thing I noticed. Your chemistry teacher was great, and that was an obviously very explosive lab.
Yes.
I noticed it in lab lessons, in that pre-lab chemistry one you showed.
Yes.
Flames were involved.
Yes. Yes. I tried to put that in specifically. Yes.
So if people can use the microphones, I had one other in the last one on the wound healing for diabetics.
Yes.
What's the final delivery mechanism for the liposome particles?
Right. So we put it in a gel. So a gel matrix that we put it in so that it stays on the wound. So it's topical. Yes.
All right. Okay. Other questions? Anyone out? It's always hard to see up here. You're all thinking about it. I know sometimes it takes a while. Come on, don't you all want the lab lessons? Wouldn't it be exciting, right? I mean, there are some right virtual chemistry labs where you can blow up the entire lab. But all right, great.
Good morning.
Good morning.
My question is, after the synthetic proteins do their job in the body, do they have a limited lifespan, and do they exit or do they remain?
Right, that's a great question. So these, they're going to remain where they are until they get, I guess, turned over. Because these materials are protein-based, they can get degraded and then they become amino acids for other proteins in your body. So they're biodegradable. And they're not going to be toxic necessarily to the body. So that's a great question.
How about the fluoridated ones?
I have a quick question. Very nice talk.
Yes.
It's a chemistry question.
Mm-hmm.
Why did you choose fluorine for your fluorinated proteins?
Yes, that's a great question. So fluorine, the reason why I chose fluorine, or we chose fluorine, was because you can use a fluorine probe to monitor the actual protein. So part of the problem of therapeutics, right, is you're gonna, even if you make an effective drug delivery vehicle, you're still gonna have to then do surgery and see if the cancer is gone, et cetera. You can't monitor this stuff. So fluorine, what it does is there's no fluorine signal in your body. But if you have a fluorine in that molecule, you can then visualize that with fluorine MRI. And what we've done, and I haven't shown you, is we can actually image these. So we can put it in an MRI. We see the image of the fluorinated protein. If we just put buffer in the solution, we don't see that at all as in terms of imaging. So now the idea, and this is what we're focusing on, is trying to do theranostics, really do therapy in conjunction with diagnosis so that you can visualize what you're doing as it's doing its therapy. So you don't have to do more invasive surgeries afterwards, and you can monitor what's going on. So that's the reason why we're interested in putting fluorine. Great question. Thank you.
Hi, I love your energy. Great talk. When you have these competitions, right, you tend to have kind of a winner and some not winners and stuff. And I know you were trying to get more women in by having kind of more social issues.
Yes.
What about the people who don't like the competition?
Right.
I mean,
okay, so...
Can some of these be formatted in a different way so that you don't always have to have winners and not winners?
Okay, so all right. So in our program, so this is for the NSF I-Corps program you're talking about. So in our program, what we do is we interview all of the students that apply, and then we actually are interested in their idea, but what we're also interested in is whether they're interested in diversity, increasing diversity. And so if there's an all-male team, for example, we ask, Did you consider another team member who happens to be of a diversity, of a woman or a different underrepresented person? And so that's how we increase diversity. But in terms of losers, all of the people that we selected get money. They get at least $350. What happens is they go through the program where they're learning about entrepreneurship, and then it's at that point where they actually submit a proposal, and they submit a proposal on a budget of what they're going to do for prototyping and customer discovery. And we read through that budget, and then we do an assessment. Like, we have a panel that does that. And then we rank those, and then we fund the extra money to those students. But all of the students who are in our program get at least the $350. So they're winners in that sense. It gets more selective as we go along, but at least all of the people, at least our students, they're all there, and they're getting all the money. They're getting at least the minimal money that we say we're giving them. Does that help?
Yeah, no, that helps.
I have another question.
Yes.
So, are these therapies available currently for cancer treatment? And if not, when do you anticipate that they would be available?
Wow, that's a very, very good question. These therapies are not. These are all experimental. These are not even, we've not done it on humans. We've done it on mouse, and we haven't done clinical trials. Those processes, clinical trials and getting to approval, take 20, even, take a long time. And so I don't foresee it being in the market until 20, 30 some odd years. So that's just due to regulations, which is really important. And that's what I anticipate in terms of that.
Although there are some fast tracks, right?
There are, but these, yes. But
there are some fast track methods for interventions that are of particular importance.
Yes, there are. You're absolutely right. These are materials, though, that have not yet been tested. It's like some fast-tracked materials for therapeutics have already passed. The materials are FDA approved. Our materials are not necessarily FDA approved. So it'll take longer, most likely, in terms of getting it to the market. All right. Thank you very much.
All right. So thank you, Jen. That was great. So one other thing, I think there's some people in the audience who've actually participated in the I-Corps program as well, right? Anybody want to raise your hands? Or anybody still? Yeah. All right. So we got a hand. We got a few hands out there, which is great. The other thing is to just let you know that it's in a way our Community College Innovation Challenge is an I-Corps spinoff, and the Community College Innovation Challenge is now open. So, and if you want to talk to the winning team last year was from Del Mar, and they are here. So, so talk to them about it, and I'd encourage you to think about coming up with a student team and actually applying to that. So thank you. And it's time for the showcase.